Hi, Team Pima. We are here in Module 5, Chapter 9. We're going to talk in this video about the neuromuscular junction, or the NMJ. So this cartoon shows what we went over in the last video. This is a cross-section of the spinal cord, and then these are two motor neurons, a red motor neuron and a purple motor neuron, and they leave the spinal cord and they travel in this cut structure called a spinal nerve. And then their axons, so let me back up. So the round part of the motor neuron is the cell body of the neuron, and then the axon is that thing that extends from the cell body and goes out and forms axon terminals here with a single muscle cell. So this is a cut single muscle cell. And then in this cartoon up top, or this diagram, is a diagram of an action potential. And we talked a little bit about that and how action potentials work in the last slide. I'm sorry, in the last video. So on the y-axis, we have membrane potential in millivolts, and on the x-axis, we have time in milliseconds. And it shows here on the graph minus 70 millivolts, that's the resting potential, aka the resting membrane potential. And that's when the voltage-gated sodium and potassium channels involved in an action potential are closed, so they're not open. And then we haven't really talked about threshold potential yet at a minus 55, but that's happens, threshold potential happens when enough positive sodium ions have moved inside the cell, causing it to become less negative, or you could look at it as becoming more positive. And at this potential, the threshold potential, now enough sodium has moved inside the cell that you can fire this thing called an action potential. So remember, at this slope of the action potential, that's where sodium is rushing in. Step 3 is where the voltage-gated sodium channels close, sodium stops rushing in, and the voltage-gated potassium channels open, and potassium, because it's higher inside the cell than out of the cell, potassium will flow out of the cell. So positive charges are leaving the inside of the cell, so the inside of the cell becomes more negative. That's called repolarization. And it doesn't say it here, but so this part of the graph is called, I can't write very well with a mouse, but I'm just going to put dipole for depolarization. And then when you go a little bit underneath the resting membrane potential, they call that hyperpolarization. So here's our cartoon again, and this is a review from the last video. So a motor unit is the motor neuron and all the muscle cells that it innervates. So the red motor neuron is innervating this muscle cell and this muscle cell. So that red motor neuron and the muscle cells together are called a motor unit. And then the purple motor neuron and all of the muscle cells it innervates, which are 3 in this cartoon, is also a motor unit. And so now we're ready to get to the neuromuscular junction. So the neuromuscular junction is where the axon terminal, so this is the axon terminal of a branch of the purple motor neuron, meets the sarcolemma of a muscle cell. So remember, the sarcolemma is also the plasma membrane, or the phospholipid bilayer of these muscle cells. At the neuromuscular junction, a neurotransmitter is released. So a neurotransmitter in this case is a chemical, and in this case it's acetylcholine. It's released by the axon terminal of this motor neuron upon the arrival of an action potential. So the action potential, which we just looked at a trace of, so voltage-gated potassium and sodium channels are opening and closing all along this purple motor neuron's axon. And when the action potential reaches the end of the axon at the axon terminal, it signals acetylcholine to be released from the axon terminal. And we'll look at some cartoons that show that. This is an actual photograph of an axon terminal at the neuromuscular junction. So here's muscle cell. Here's a muscle cell. And there's a nucleus. Here's another muscle cell up top with a nucleus. And then this is the axon terminal. So this area would form a neuromuscular junction. And the number of muscle fibers that you could innervate, so remember the muscle fibers are also called the muscle cells. So the number of muscle fibers / cells innervated by or per motor unit ranges between 4 and 300. So in this cartoon, the purple motor unit, you have the axon of the purple motor neuron innervating 1, 2, 3 muscle cells, but you could innervate potentially up to 300 muscle cells with just one motor neuron. So here's the cartoon of the neuromuscular junction. So we've been looking, this one up in the top right, we've been looking, here is where the neuromuscular junction is. And then there's a different cartoon. So this is the axon. Encased in this blue thing is a myelin sheath, but the yellow thing is the axon of the motor neuron. And these little ends here are branches of the axon, and they form a neuromuscular junction. And then you can see they take this black rectangle cut and they bring the arrow down here to show the more detail of the neuromuscular junction. So this yellow thing, this is the axon terminal of the motor neurons, axon, and this is all the stuff inside. So one thing you can recognize probably, there's a mitochondria, so obviously there's some ATP needed here. So the mitochondria lives in the axon terminal, and then you also have these synaptic vesicles, and these little green balls inside these vesicles are acetylcholine. We talked about how acetylcholine is released from the axon terminal. And then the axon terminal, which is the yellow structure in this cartoon, doesn't actually contact the plasma membrane of the muscle cell. So here's the plasma membrane of the muscle cell. All of this that I'm tracing, it's all plasma. It's actually called the sarcolemma in muscle physiology, but it is the plasma membrane or the phospholipid bilayer. And you can see between the axon terminal in yellow and the sarcolemma, or the phospholipid bilayer of the muscle cell, there's this thing called a synaptic cleft. So it's a gap or a cleft between the neuron and the muscle. And the cleft, the synaptic cleft, is filled with interstitial fluid. Okay, so we talked about how the action potential, so the action potential, opening of voltage-gated potassium and sodium channels in succession, the action potential arrives at the axon terminal of the motor neuron, and what that is, it's a change in electricity. So the action potential is changing the membrane potential of the axon, and what happens is that's an electrical change. And these channels here, these are voltage-gated calcium channels, and they sense that change in electricity that the action potential brought to the axon terminal of the motor neuron. And these voltage-gated, so these are voltage-gated calcium channels, they open, and calcium rushes in down its concentration gradient from the extracellular fluid into the axon terminal of the motor neuron. And then what happens is a whole bunch of biochemistry that we won't talk about, but this calcium causes a series of molecular events that allows for the vesicles in the axon terminal to release these little green things, called acetylcholine. So acetylcholine is a neurotransmitter. Okay, and then so you can see we've been doing steps. So the previous slide was step 1 in the neuromuscular junction events. This is step 2. So calcium ion entry, and actually I just talked about that in the last slide, but calcium entry, bunch of biochemistry, causes release of acetylcholine into the synaptic cleft. Then what happens is acetylcholine, these little green dots, so acetylcholine is now in the synaptic cleft, and acetylcholine will diffuse across the cleft and bind to these acetylcholine binding sites on these purple channels. And these purple channels are called ligand-gated sodium receptors. So remember, we've talked about voltage-gated sodium channels. These are ligand-gated sodium receptors. So this gets a little tricky, but the ligand in ligand-gated is acetylcholine. So the ligand is just a general term for a molecule, but so the ligand acetylcholine will bind to these ligand-gated sodium receptors or sodium channels, and that's going to cause these sodium channels to open. But this is really tricky, and if you make a note of this, there are lots of different types of channels, but there are voltage-gated sodium channels, but in this cartoon we are talking about ligand-gated sodium channels. And I think I would rewrite this as a sodium channel instead of a sodium receptor so that we can make a list of our channels and become channel physiologists. Wouldn't that be fun? Okay, so what happens when acetylcholine binds to these ligand-gated sodium channels? Well, they open, and that means when they open, that means that the plasma membrane or the sarcolemma becomes temporarily, as long as the channels are open, plasma membrane becomes temporarily permeable to sodium. And sodium is high outside the cell and low inside the cell. So sodium rushes into the cell. And these channels are a little tricky because as the sodium is rushing in, some potassium can also leave through that same channel. But more sodium is rushing in than potassium is leaving. So that positive, remember sodium is positive, so that's going to make the inside of the cell more positive as positive sodium rushes in. And you'll see that depolarization. So I showed you that in the action potential where sodium is rushing in, that first slant. I think I have I don't have a graph of it. Oh, I should have put a graph up. Well, anyway, the depolarization of the plasma membrane because of sodium coming in via the ligand-gated sodium channels, this whole area will get more and more positive. And then this is tricky because you have ligand-gated sodium channels just underneath the axon terminal, but here on the periphery of the neuromuscular junction, just outside the neuromuscular junction, you have sodium channels as well, but they're not ligand-gated, they're voltage-gated. And so if you come up to the top cartoon here, it shows, here's the axon terminal of the motor neuron, here's the sarcolemma, or the plasma membrane of the muscle cell, this blue thing, and then here's a mitochondrion. And in this part, when you're outside the neuromuscular junction, so you're not right underneath the motor neuron axon, you have voltage-gated sodium and voltage-gated potassium channels. This guy is a ligand-gated sodium channel, and this green dot is binding acetylcholine. So as sodium rushes in the ligand-gated sodium channel, this area gets more and more positive. As this area gets more and more positive, you get more positive sodium rushing in. That's going to activate these voltage gated, because they're going to sense the change in voltage. So remember, the resting voltage is minus 70, or some negative number here. As positive sodium comes in, these guys are going to sense that the membrane has gotten more and more positive, and because they're voltage gated, that means they respond to a change in voltage, they're going to start opening, and now opening and closing, and now you can have an action potential run down the length of the muscle cell. So how does the muscle contraction start or stop, excuse me? In the synaptic cleft, you have a molecule or an enzyme called acetylcholine esterase, and it breaks down acetylcholine. It breaks acetylcholine down into acetic acid and choline, and then muscle contraction will stop. Okay, so a couple other notes here. When acetylcholine binds to the ligand-gated sodium channel and sodium rushes in, making the inside of the cell more positive, that when you use a ligand-gated channel to change the membrane potential, that's called a local potential. When you use voltage-gated sodium and potassium channels out here, outside of the neuromuscular junction, that's called an action potential. And then the muscle cell will return to its normal resting state when the sodium-potassium pump reestablishes that concentration gradient of sodium and potassium inside and outside of the cell. And then so we've talked about different channels. These potassium leak channels, they're always kind of working, and potassium's always kind of leaking out just a little bit, but not enough to really affect to create a local potential or an action potential. And this cartoon is an analogy comparing the action potential we just talked about to a flame consuming a dry twig. So here's the small twig. Here's the match. The flame ignites the twig, and then the flame spreads rapidly along the twig. That's like, if you are thinking about action potential graphs, so remember you have millivolts on the y-axis and time on the x-axis. So you have resting potential, let's say at minus 70, could be minus 90, depends on the book that you're looking at. So you have the resting membrane potential. And the ligand-gated sodium channels are open. Sodium's coming in slowly, and then at minus 55 millivolts, which is threshold potential. Boy, it's hard to do this with math. At threshold potential, so all of a sudden, boom, the match ignites the twig. Yay, you get an action potential, and then you come back down. So that's what this analogy is supposed to help you understand. And then this is just a cartoon that shows everything that we've talked about, just in kind of all in one slide. So I think that's it.